Hello, I'm Shobit and today I'm here for my topic presentation on identification of bacteria. What is identification of bacteria? Once a bacterium has been obtained in a pure culture, it has to be obtained. There are different techniques for identification of bacteria. Out of those techniques, staining technique is one of them. We are going to use simple staining techniques and later we are going to use different kind of techniques in that. Firstly, we are studying the simple staining procedures and its mechanism. In simple staining, is a method of staining in which bacteria are stained by using a single stain. When we are going to stain the staining technique, we are only using the one type of stain. Okay. Simple staining is also called as monochrome staining or positive staining because it is also known as a monochrome staining because it is only using one stain, and in which we are using positive staining because it will give positive results. Examples of simple stainings are methylene blue, sapphire, basic fusion, crystal solids, etc. Let's move around the second slide, which is the procedures for the simple staining. Firstly, we have to take a clean grease-free slide. Later, we have to wash it, and later we have to wrap the glass slide by a brown paper or a silver foil. Later, we have to put it under the hot air oven at around 20 degrees Celsius for around 40 to 45 minutes. Later, we have to prepare a smear through a nylon loop, sterile loop, basically. Later, we have to air dry the smear by spirit lamp. We have to flame the glass slide 3 to 4 times in this procedure. Later we have to heat fix by passing the slide through the spirit lamp. Then we have to flood the stain by 3 minutes. Basically we are going to flood the smear for flood dye. Later we have to gently wash it around the running water. Later we have to air dry the smear which we have stained in the wash basin under the microscope. We have to study which type of bacteria which are produced in this procedure. Later on we are going to study about the applications for simple staining. Simple staining colors the bacteria, make them easy to observe under a microscope. It is mainly used for studying morphology of bacteria, their size, shapes and arrangement. In simple staining procedure, basically we are going to study about the bacterial smear, the size, morphology, their arrangement and shapes. For the latter procedure, we are going to study around the gram staining procedure. For the gram staining procedure, which was discovered by the scientist microbiologist Hans Christian Grams in the year of 1884. He classified the bacteria into two groups, which are the gram-positive bacteria and gram-negative bacteria. The difference is based on whether cells retain or lose the primary stains after treatment with a decolorizer. In decolorizer, we are going to use around 95% of alcohol. Gram staining is one of the most widely used techniques in the microbiology. Several modifications were made later by the Huggers and Gons methods because they give the fantastic observations and results. Let's move around the next slide, which is the gram staining procedures. For the gram staining procedure, it is basically same as of simple staining. Firstly, we have to take a clean grease-free slide. Later, we have to put it under the hot air oven, wrapping it through the brown paper or silver foil at 180 degrees Celsius for around 40 to 45 minutes. Later, we have to heat and dry the smear. Later, we are going to steam the smear by crystal solid for around 1 to 2 minutes. Later, we have to wash it under the wash basin by tilting mechanism. Later, we have to add a gram iodine solution for around 1 to 2 minutes because the smear contains bacteria which includes mycolic acids through which they will add to and stick to the glass slide. Then we are using the decolorizing agents as alcohol. Later we are going to wash it around 2 to 3 minutes later. We are going to counter stain these meal by saffron or basic fusion for around 1 to 2 minutes. Later we have to observe the ground stain of the bacteria under the microscope for the viable results. Basically, ground stain procedures includes two types of results. If the results are positive, the organisms or the microscopic organisms which are present over the glass slide will gives a violet results. Violet color bacteria we are going to see under the microscope, and later for the gram-negative cells, pinkish color is obtained. Later, we are going to study about the functions of reagents in gram stain, which includes the crystal violet, gram iodine. Crystal violet we are going to use around its primary stain and stains for all the cells. And for gram iodine, we are going to see about the moderate or class CV1 complex strengthening stain binding agents. 95% alcohol, which is ethanol also, we are using it because it gives the decolorizing lipid solvent removal, which is the CV1 complex from gram-negative cells. Later, saffron and basic fusions are used as a counter stain which gives pinkish color or to gram-negative cells. Thank you.